Good afternoon, everyone. So the Biochemical Society and the Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. So this series includes various topics covering research areas of molecular biosciences, as well as also practical sessions in order to support your career development. So each of these webinars will give you the opportunity to ask questions at the end via text, so using the chat box in the app. And we also welcome suggestions for future topics and speakers to feature in our webinar series. So please see our website for more details about that. So I'm Charles Winterhalter. I'll chair the session today, and so I'm a recently appointed group leader in Newcastle University in the UK on the Wellcome Trust Early Career Award. And as it's the focus of today, our today's webinar will be from postdoc to group leader, starting your own lab. And so it is dedicated to our early career research program of webinars. So in this session, our invited speakers will discuss their experiences of starting their own independent research groups and also share some insights to equip you with the best knowledge to start your own career, your own lab, and achieve these milestones. So before I hand over to the first speaker, I'd like to mention that questions will be asked at the end of the webinar, but please do feel free to send your questions during the talks and we'll walk through that at the end of the webinar. So if you have a question, please please type it in the question box that you might see on the right of your screen, as shown on the screen sharing option now, stating who your question is for. Don't forget to state that. And we'll try to answer as many as possible at the end. And without further ado, our first invited speaker today is Clinton Lau from the University of Oxford. So Clinton leads a lab to investigate the cytoskeleton of the malaria parasite, Plasmodium falciparum. So his research career began with a DPhil at the University of Oxford, where he studied host pathogen interactions involved in Plasmodium falciparum in the lab of Professor Matt Higgins using X-ray crystallography and biophysical technique. So he then completed a postdoc position in the lab of Dr Carter at the MRC LMB in Cambridge, before successfully applying for a Wellcome Career Development Award to establish his lab somewhere at the intersection of his DPhil and postdoc studies, starting in late 2022. And with this, I'll hand over to Clinton.
Brilliant.
I can see your screen, no problem. And
Fantastic. I can. Good. Okay, so well, thank you for the lovely introduction, Charles. It's an absolute honor to be speaking at the Biochemistry Focus webinar, particularly on this topic, because when I started my own lab, I got a lot of advice. So it's nice to be in the position I can share my journey with the hope that some of it will be useful for you guys listening too. I'm going to talk today a little bit about my career journey. I'll focus in on taking the step to group leader, particularly on the sort of how did I decide research direction sort of topic. And then I'll discuss a couple of issues that came up during this process, particularly battling introversion and social anxiety, which I continue to do, and also having a child, which I guess I also continue to do. Finally, I'll give a couple of perspectives now that the lab has been going for just over 2 years, particularly about things that things I did, things I'm glad I did, and things I might do differently next time. At this point, I'll acknowledge just one person from this list, which is Grace Frankel, who's my wife. I'll acknowledge the rest at the end, but without Grace, certainly I wouldn't be in this position. And something I should note for these sorts of talks is there's going to be a heavy element of survivor bias, because obviously both myself and Clara, who you'll hear from next, are group leaders at the moment. So just take this talk for what it is. It's my journey. Hopefully some of the advice will be useful to you. I have to say I'm not quite arrogant enough to say the speech will be inspirational, and I don't yet really consider myself successful. Anyway, so my career journey has actually always been in the UK. I grew up in Leeds in the north of England, and then did an undergraduate at the University of Oxford. At that point, even as early on as this, so I met my wife, then girlfriend. We decided for family reasons, primarily we wanted to stay in the UK. So my next step from there was to apply for a PhD which, as Charles mentioned, I did in Oxford at the Higgins lab, looking at malaria host pathogen interactions. There I am doing my PhD. This was followed by a 7-year postdoc in a very different biological field, but using similar techniques. So I went to the MRC LMB over in Cambridge to work in Andrew Carter's lab to study the microtubule motor dynein. In both cases, through my PhD and postdoc, I was developing skills in structural biology, biochemistry, some biophysics as well. Now, during my postdoc, our child was born, again, with thanks to my wife. That was 2 years in. And then skipping forward to January 2021, so we're now looking just over 5 years in, is when I was starting to make the transition out of my postdoc life. At the time, I started applying for fellowships and jobs, and what we'll see at the bottom here is it took almost 2 years before my next position as the head of the Lau lab was started. So there is a bit of a time lag there. Let's fill in a little bit of the details between January 2021 and November 2022. So, of course, I was doing some postdoc work at the time, but in addition, I was applying for these fellowships and jobs. I spent about 8 months in total looking for a fellowship sponsor. So that's an institute that will sponsor my applications. I reached out to both Oxford and Cambridge because they had everything I needed for my future research. But in the end, Oxford were gracious enough to allow me to apply with them as sponsor. And then on the fellowship side, I went through 3 different fellowships, the Wellcome Career Development Award, the ERC Starting Grant, and the MRC Career Development Award. At the same time, I was applying for jobs as well, of course, applying for 3, I think it was in fact 4 rather than the 3 I've listed here. And what you'll notice is that actually of all the applications I did, and I only did 6 or 7, only one of them was successful. So the success rate was very low. This is quite typical, and I would note that actually my journey between starting to apply for the next step and actually achieving the funding that I needed it's on the shorter side of average when I speak to other people who've undergone similar journeys. So that's just something to note. What we'll do next is we'll take a trip back in time to this time period here, to January 2021, when I was starting to apply for fellowships and jobs. And now at the time, I was certainly worried that I would be somewhat penalized for staying in the same country, indeed just shuttling between Oxford and Cambridge, although of course I understand that I have the massive privilege of being able to do this in the UK, but I was still worried that it would penalize my applications. And also I was a little bit concerned on future research direction because I felt both my PhD and postdoc fields were relatively saturated. Of course, the academic journey in terms of staying in the UK, I can't really do much about. I just had to sell it as much as I could. However, the research direction I could do something about. And so I took a good amount of time prior to starting to apply for these things, deciding on my future research direction. The criteria I had for my research idea, I mean, is nothing new. I wanted it to be novel, of course, distinct from my previous works in both my PhD and postdoc, but not so distinct that funders would think it unrealistic, me wading into a completely new field, let's say. I also wanted to enter an academic field where I saw there'd be space for my lab to grow. I envisaged at the time my lab would be primarily structural biology, biochemistry. I thought maybe I'll go into a little bit of cell biology as well, but primarily structural biology and biochemistry. In terms of where the idea came from, I had this question 2 years into my postdoc. Obviously, coming from a malaria lab into a dynein lab, I did wonder, what does dynein do in malaria? I didn't think too much of it at the time because I had other things going on, but I revisited it when looking at my future lab direction. And it turns out we don't know a lot about what dynein does in malaria, I would say. And so definitely from a narrative arc, it seems on the surface that I've married the two, my PhD and postdoc, I expanded it, of course, into more cytoskeletal organization of malaria parasites. So now I run a lab looking at how microtubules and other filamentous systems contribute to the malaria parasite organization. However, this did actually involve moving into a new field, which we'll call the Plasmodium and related parasites basic biology field. And in this regard, all of the researchers I interact with nowadays, very few of them, I didn't know any of them actually during my PhD. I would consider them almost separate fields compared with malaria host interactions. I'll speak a little bit now about how I developed this initial idea moving forwards. Because I considered that I was moving into somewhat of a new field, I thought that collection of preliminary data was important. And here I really have to thank Professor Catherine Merrick at the University of Cambridge for giving me some parasite material that I could probe and look for microtubules, essentially. And also thank my former boss, Andrew Carter, for giving me the time in his lab to collect this preliminary data alongside my postdoc work. I think the collection of preliminary data was important for my funding application because of this field transition. And also at this point, I think I did more reading than I've ever done at any point in my life ever. This was helped for me by starting mind mapping a lot more than I'd done previously. So I would suggest this software, FreePlane, which is a mind mapping software I still use, and this really helped me organize thoughts and ideas around in my head before committing to writing a particular application. I also, during this application writing period, really found out how I wrote. I get this question quite a lot from postdocs who are looking to apply for these big grants in terms of how do you write such a big application. It's definitely very personal, and so to share my experience, I initially struggled on getting the ideas down on the page to begin with, because I think I was too worried about expressing the idea perfectly. What I would suggest, and what certainly helped me, is just spend a few hours just putting down ideas on a page without caring that any of it makes sense. It sounds weird, and I'm too embarrassed to show any of my initial drafts to anyone anymore. But after that initial stage of essentially word vomit, I then went through a lot of rounds of editing to curate that into an application that I then sent off to the funders. The other pieces of advice I would have at this stage is get a good idea of the timelines for all of your deadlines, particularly if you have collaborators or perhaps host departments that will need to see and perhaps write sections of your applications. Contact them early and frequently to make sure they get enough time to write the bits that you need. And finally, I would advise that you talk to a lot of people about your application and your idea generally. I can say that I talked to at least 15 to 20 people during this process. Some of them, of course, were to read my application in its various draft stages. Some were to give me mock interviews, but I put a lot of effort actually in talking to people already in the Plasmodium basic biology field, and they were very receptive to another structural biologist coming in to explore. And I think that was also important when it came to funding, that I'd already contacted people in the field I was transitioning into. This last point, talking to a lot of people, is something I found very hard. And I'll talk next about a battle that I've had and continue to have, which is a battle against social anxiety and introversion, because it's something I don't hear often talked about in these sorts of fora. When I feel particularly socially anxious, I remember the quote I have at the top here, which is "Don't ask, don't get." It was said by one of my friends, Girish Mali, who's a group leader at the University of Oxford. But yes, I would class myself as introverted. I still get socially anxious pretty much daily. And I know that a lot of PIs feel this way too. So when I was a postdoc, I had the idea that every PI was super confident, or at least seems it, super extroverted and whatever, but this is not the case. So you are certainly not alone. My battles specifically are around public speaking presentations and also speaking to new people and people I perceive to be important. I've got a couple of bits of advice to dispense if you too are socially anxious or introverted. The first is a little bit unfortunate, is at least for me, the nervousness before presentations has never gone away. The reason I say this is because during my academic career so far, I have been hoping and planning for this nervousness to go away at some point. But since I realized that I'm going to get nervous no matter what, I have focused more on developing strategies to cope with the nervousness that inevitably comes before presentations. The things that particularly have helped me is firstly practice, practice, practice. And I still hear other group leaders as well practicing their presentations before. I was practicing this one this morning and a little bit last night. And it really helps me be confident in what I'm saying and hopefully confident that I will hit my time. Also, something you can do is some science outreach. Because a large part of the job as a group leader is to sell your research, of course. And I can tell you there is no audience harder to sell microtubule motors to than families who are walking around a science fair on a rainy Saturday in Cambridge. And so if you can sell your research to some of these more challenging audience, you can definitely sell them to funders. In terms of speaking to new people and asking for help, again, I would say it's never gotten completely easy for me. I still get a lot of anxiety around this, but of course it's super important for pitching applications to jobs or fellowships. And also when you get into the field, it's going to be important for making those scientific connections, in part which makes science very enjoyable, if I'm honest. A couple of things that I have done that I continue to do that help me. Firstly, I quite rigorously reputation check before speaking to people. This will be through people that I know. I might ask, is Professor X, are they very friendly? Are they very nice? Is there anything I need to be cautious of? And that helps quell some of the anxious feelings that I get. And the second thing, particularly when emailing someone I don't know, is I try and remember that the worst they can say is no, or they're too busy, particularly regarding sort of advice for these fellowships applications. And to be honest, I've never got the answer no. Sometimes people are too busy, but it's very seldom that this happens. People are usually kind. The next issue I thought I'd bring up in this sort of forum is having a child in academia. And when I was a postdoc, I went to this sort of early career forum, and Shaw Sherez, who is a group leader at the MRC, said, there is no good time to have a child in academia, but there are worse times. At the time, my wife was pregnant and I was feeling very nervous about what was to come, so this helped a little bit. And similarly, I hope if there are any of you there who are trying to plan a family, this might be a little helpful at least. At this point, I will definitely acknowledge a couple of privileges I have in terms of I am the secondary caregiver and also a man with a supportive wife who's not in academia. But I am completely dedicated to spending time with my child, and it's something I enjoy a lot. I also, at the time, I had a very supportive, understanding boss when we had our kid during my postdoc. And now I have a very understanding lab when I need to go off and pick up my child from school or take them to dentist appointments. My lab is very flexible about this. A couple of things that I would get ready for that I perhaps wasn't completely prepared for. Your lifestyle will completely change. I mean, this sounds very, very obvious, but I can't, and more importantly, I won't now spend as long as in the lab as I did during my postdoc before I had a kid or during my PhD. As a result, I've had to become a lot more efficient, which is going reasonably well, though there are costs which I'm happy to talk about if you're interested. There's also a long period of adjustment after you have your kid, particularly after you have your first kid. In our case, I would say first full 2 years were really rough on the sort of home side. We're doing the best that we can, of course, but just things to say, if you're having a particularly rough period after having a child, it is very normal, and do reach out for help if you need it from all of the various support systems that you might have. A quick note on childcare. I knew it was going to be expensive. I didn't know quite how expensive, but it is still very expensive, at least in the UK, and there are very long waiting times as well. Again, a bit of perspective here. We got on the nursery waiting list before our child was born, which felt a bit weird, if I'm honest, but just to flag that in case you're not aware. And finally, something that I still battle with, I currently feel guilty in 2 ways, which is great. On bad days, I will feel guilty either for spending time working, but equally spending time not working. That is something that I manage well on my better days. It plagues me on my worst days, but again, is kind of par for the course and something to get ready for. Okay, I'll now focus a little bit, now that my lab has been going on for just over 2 years, on a couple of perspectives. So as I mentioned, my lab's been going for 2 years now. About half a year ago, we had a lovely visit from a journal editor who will remain unnamed. And they came for a meeting in my office and said, this lovely thing I have up top here, you haven't got anything to publish yet, question mark. That hurt a little bit because yeah, we've got stories in the pipeline, but nothing ready still just yet. It did send me into a little bit of a personal crisis of confidence, but after looking back, I can happily say that I don't regret anything so far. Again, it's very normal and I still have good days and bad days, but ultimately what drives me as a group leader is I love discovery science. I really, really love it and see its value. And also as a group leader now, I really enjoy managing and developing people inside my lab, but also people I meet in the institute, in the country when I go to conferences, all this sort of stuff when I can. And these two things have kept me going in academia, and I think they'll see me afar. To give you a few things that I recommend that you would do when starting your lab that I did do, introduce yourself to a lot of people. For me, obviously, that involved overcoming social anxiety, etc. But I'm glad I did so because you never know where the next useful piece of advice will come from. I spent a lot of time before I had people in my lab working out systems to minimize my lab's individual workload. Things like communal ordering, communal lab books, rotas, etc., as well as a good sort of lab introduction document. These help efficiency in the lab a lot. And I also spend a lot of time thinking hard about what qualities I need from my lab members. Particularly worth flagging here is I ask all of my lab members if they feel like something could be improved to tell me as soon as possible, and they've been very good for that, and it's helped us grow as a lab and move forward. And the last thing I'd recommend that I did do is a lab leadership course, because I wasn't trained in lab management beforehand, before this course, of course. And I'd recommend the EMBO course. Though it is expensive, I'd say it's worth every penny. A couple of things I'd recommend doing that I didn't do. I try and seek more training on the budgeting side if possible, though that's not always the case. Now I have to get it sort of piecemeal through experiences of other group leaders and of course with our financing team, but I wish I had a little bit more training on that before I wrote my applications. I'd also try and budget in some administrative support if possible. And finally, for each fellowship application, I'd recommend getting a couple of successful ones, because every fellowship and job, I suppose, is a little bit different. And that's something I didn't do, which I could have done a little bit better, I think. And that's me. There's some other resources here, but for sake of time, I won't go into them too much. And again, to acknowledge the people I've talked about in this talk, particularly my wife. Thank you very much for listening.
Thank you very much, Clinton. That was actually inspiring, although you didn't think it would be at the start, but that really was. So we'll move on to the next invited speaker, who is Clara Correia-Melo from the Leibniz Institute on Aging. So Clara recently started her group as a group leader in Germany, and her research focused on biochemical crosstalk of aging cells. So Clara has got a background in human pathologic anatomy. She did her PhD in the molecular biology of aging at the great University of Newcastle, whilst holding a GABA fellowship. So she then carried out a postdoc at the University of Cambridge and the Francis Crick Institute, and then the Charité Medical University Berlin in Germany, while exploring the metabolic interactions of microbial communities. And with this, I'll now hand over to Clara. I can see the slides. And can I hear you?
Yes. Can you see now the slide? Sorry. Yeah.
Perfect. Yes. Thank you.
Awesome. Thank you very much, Charles, for this nice introduction, and thank you to the Biochemical Society to invite me to share my experience on starting my own lab. It has been a journey, a very, very fun journey, but also challenging. I would always like to characterize it more fun than challenging. So you will see throughout my presentation how I balance that challenge and fun aspects. So this is, I mean, you can see here in this first slide, my very empty lab. So some people won't face this challenge because they might go into an institute or university that has already everything what they need. I actually had to equip this lab, but I'll talk about all these things later. I am also very glad that Clinton covered things that I'm not going to talk. So I do not have kids, so I'm glad that he mentioned that. But I can say that I'm going to talk about starting your own lab from a German perspective, not a German perspective, but starting it in Germany. And I can tell that the support for scientists and people in general that start a family is great in Germany. So there is a huge support. There is a 1 year paid paternity or maternity leave. So just to get this bit covered, if you have any questions, then you can always email me, which I have at the end of this talk. But now, starting with my presentation, I would like to give you an overview of my talk. So I'm also going to give an introduction of my journey and my lab, and then I will tip into considerations and pitfalls to avoid, of course, always from a very personal perspective, but also from what my colleagues also share, what I see my colleagues also talking about, and so on. So then an important aspect also of starting your own lab is securing grant funding and networking, but also I'll provide some general insights into starting your own lab. So my journey, I'm actually originally from Azores, a tiny group of islands in the middle of North Atlantic that belongs to Portugal. And then I started by getting a diploma in human pathology in Porto, in the north of Portugal. Then I started doing research as a research assistant in a lab, also in Porto, that was focused on cell division and aneuploidy, and how aneuploidy was intimately linked to cancer development. And there I mostly used mammalian cells and mouse model systems to answer our questions. After that, I moved to Newcastle University, as Charles has a great university. So Newcastle University was very focused on aging research. I think it still does, but at the time it was really strong on aging research. And I wanted to understand why we age at a molecular level. And I was particularly focused on how mitochondria was important for the pro-inflammatory phenotype of senescence, held as aged cells. And for this, I studied senescence in mammalian cells, but also in mouse models. Then I completely changed and I moved to Cambridge for my postdoc, but the lab then moved to the Francis Crick Institute in London, where I did most of my postdoc. And at the final year of my postdoc, the lab moved to Charité Medical University in Berlin. So I spent my last year of postdoc in Berlin, just mostly writing up my papers and applying for positions. There I changed completely. I studied metabolism in yeast, mostly from a chemical point of view, from a metabolite point of view. And for this, we used metabolomics, but also proteomics and systems biology to understand how cells communicate chemically and how this impacts cellular physiology. And since 2 years ago, since February 2023, I am a junior group leader at the Leibniz Institute on Aging, and I lead the host microbiome metabolism in aging lab. And there we combine almost all these different aspects of research that I studied previously, so both cellular aging, metabolic communication at the chemical level, also mammalian cells, microbial cells. So it's a combination of many things that I actually studied before. And more recently, I became an associate group leader of a cluster of excellence called Balance of the Microverse. And I can talk a bit more about this if you have some questions, but this is a big governmental funding initiative in Germany, which basically involves like 70 groups, sometimes a bit more, sometimes a bit less, that focus on a specific topic. Here is the balance of the microverse, and this is a unique opportunity to really drive innovative science because everyone studies this overarching theme from a very different perspective. And it was great to actually become part of it. And so now going into what we do in my lab, as I mentioned, I then combined many of the things that I studied before. And so the big question in my lab is really, how does the chemistry of one cell alters the chemistry of the neighboring cell and overall cell population physiology and responses? And we study this during aging. And because of that different background, scientific backgrounds that I just had as part of my path, so we can study these chemical interactions not only in the host, between host cells, but also in between cells in microbial communities. But a big focus of my lab is actually study how microbial cells and host cells communicate chemically, and then how will this influence how the organism ages and how cells will respond to different stresses, but also drugs. And so the reason why I'm showing now what this entails is because this was really important to set up my lab. So we use high throughput approaches. We use omics, particularly metabolomics, and I had to set this up in my lab. We also use proteomics. This is actually part of a core facility we have in the Institute. Phenomics, we collaborate a lot for metabolic modeling. And we use all of these to understand how this shared chemical space influences cellular physiology and responses. Right. Now, in order to tap into what do we need to consider, or what I would say we need to consider and the pitfalls to avoid when starting your own lab, even before going to the where question, I think that getting a mentor or several mentors is really important. So I wouldn't consider myself as an introverted person. I tip more into the extroverted person. So I do reach out. I also have in mind that the worst I can get is a no. So, and I do understand if you are an introverted person, as Clinton mentioned, these can be a barrier, but the importance and how helpful is to get advice from a person or several persons, it's very important. If you do get a mentor, it's not that you're just talking to random people. Usually a mentor is someone that you feel comfortable talking about any issues or the advice or questions that you have. So managing to find those mentors are really important because they've gone through the process and they will be willing to help you. As Clinton mentioned, I never got a no, and most people the maximum they said was, yeah, I'm a bit busy. So do get a mentor. And then where it's a really important question when starting your lab. So it's really important that you set your research apart from the existing research at a specific university or institute that you want to apply or that you want to be part of. So be an asset, not competition, meaning that the research that you will do in that institution needs to be relatively different, ideally quite different, but in a way that it complements and it benefits to the research in that institute. Because if you're doing more of the same, then you'll be already at a position of competition, and this will make things harder at establishing yourself as in your own lab. So make sure also that the environment at that institution is supportive. Even when in cases when you're not competition, that you actually complement the institution where you then get your position, then you need to make sure that this institute or university is supporting you at multiple levels, equipment level, also at management level, at administrative level, financial level. There's many different areas where you will need support. And this is really important, that you make sure that wherever you go has a very supportive environment. This will make things much easier regarding starting your lab. So then there's the question, university versus research institute. So there are very, very different aspects to consider at the university and the research institute. There's pros and cons in both of them. And again, I'm mostly talking now, I mean, this is also valid for the UK, but also talking mostly in Germany. So there is something at universities that is called the tenure track, which means that after your probation period, usually 5 years, then you can get a permanent position as a professor at the university. This is quite common now in most universities, and there's a security feeling to it that after this period you don't have to move again, you don't have to find a job again, a position again. On the other hand, this usually entails quite a lot of teaching duties, and if you're starting your lab, this is quite distracting, which usually doesn't exist at research institutes, or when it exists, it at a much lower level. So your starting package also is very important. So when you're starting your lab, there's something that is called starting package, which means that it's the amount of money that you are given per year for consumables and what's not, the amount of money for equipment, and also the number of people and which categories of what PhDs, lab manager, or postdocs, how many of these you get, how many of these lab members you will get. So at the universities, usually the starting package are much lower. So maybe you get 1 or 2 people. Most of the times you don't get specific funding for equipment. Usually either there is already there, the equipment, or you will have to apply through external grant funding, third funding parties. But so in the starting package, so it always considers really pay attention for your starting package, because this really will make your life either a bit more difficult or easier. It won't stop you, but it can make things easier. So usually research institutes have better starting packages, but what is the disadvantage of research institutes, particularly, I guess, mostly talking again in Germany. So we have Max Planck institutes, we also have Leibniz institutes, Helmholtz institutes, and there is another one, but these are the major research institutes in Germany, and most of them don't have tenure tracks. So you have a position for 5 years with an extension of 4 years in most cases, but then you have to go. You have to pass through this process all over again. You have to apply for a position, you have to secure this position, hopefully now in a tenure track or as tenured, and this puts a lot of people away. So the starting package is good, but there's no security regarding a permanent position. Research institutes usually also have a better financial and administrative support. But again, it's what you feel more comfortable with, the security of a tenure track or all the support to start with. I went for a research institute because, I mean, the starting package was incredible, and all the support that I've been having so far has really been amazing. And I think this makes starting your lab very, very different and easier. Then is the question of who. So I guess my talk is focusing mostly once you're starting really your lab rather than before. So here you already have your lab, you secured a position either at a university or at a research institute. And one of the first things you need to do is hire people to then do the research in the lab. And this is one of the most challenging things. So finding and recruiting skilled people and self-driven people is very important and can be hard. Sometimes people are skilled, but they're not self-motivated, and it's easier to train someone than motivate someone. So really at the interview level, and also it's really important that you pick the right people. There will always be mistakes, and you learn from it. Whenever I got advice from my mentors, they always said, no matter what, you will always hire or have some lab members that are a bit more challenging than others. It's part of it, but you will see what is best for your team with time. So this is something that comes, but that's a very important aspect. And also managing early expectations. So one of the things that I did, or was a mistake in the beginning was not making it clear what is expected from each of the lab members, what's expected from a PhD student, what's expected from a postdoc. And because I thought this is common sense, I thought that it was clear as a postdoc that you drive your project, that I am not driving the project, that you should drive. But this is not clear, and some of this stuff are even in the contracts. But so it's very important that you sit down, that you talk, and that you make sure that communication is effective, that both people are on the same page. And then the different personalities of the different lab members is something that to me is the most challenging thing as a group leader. It's very fun, and it's incredible. I guess only now as a group leader, you all, of course, you are in a lab, you see the different personalities, but you don't have to manage these different personalities. So it doesn't mean that there's bad or good personalities, they're just different and they see things different, which is amazing, but it's also challenging. So I think it's important to be aware of it and to bring the best of each personality and help each personality to overcome their downsides if they would like to, right? So the next thing is how to transform an empty lab into a functional lab and not everyone will be in this situation, but some people will bring completely new research to their institute. And this was the case of my lab. So we bring high throughput technologies, we bring metabolomics, and this was something that didn't exist in my current institute. So it's been incredibly fun to arrive to a completely empty lab where I was alone in the lab and transforming it into something completely full of technological platforms. And now I have a team of around 8 people, and the lab is full and buzzing. And this has been an amazing journey and fun. But what does this mean to go from an empty lab into a fully functional lab? So if you do have to equip your lab, then you have to start as early as possible. So I started actually 4 months before I started at my institute. So you need to start talking to vendors. The equipment we have is quite specialized, this high throughput robotics. So I talked to many vendors. Then you have to write tenders. Tenders are big documents that then become public and where companies bid, and then you will go with that company that has better price and so on. So this takes a huge amount of time and effort. Coordinating internally with the building construction management team. So it might be that they need to even hire teams to come to the institute to do adaptations to the lab so that you are able to have the equipment that you need to do your research. Again, this takes time and requires coordinating and a lot of talking, a lot of meetings. Delivery times, it takes time to get tender process, have specific times, and then you do the ordering, and then you have the delivery time. So all of these takes a huge amount of time, making sure it works, getting training on this equipment and maintaining. So these are just quite a few of the things that you need to deal. That it's not science, but it's all the things that you need to deal when you start your own lab. Just to give you an idea, I started in February, and then I actually started talking to vendors 4 months before. And this is the timeline. So we got our latest equipment, an anaerobic chamber, in June last year, so a year and a half after I started. And this anaerobic chamber was only fully operational in November last year. So nearly 2 years after. So if someone comes to you like they did to Clinton and ask, where are your papers? Don't feel bad about it, because things take time. It takes time to establish a team, to establish your lab. So it's what it takes. But I think if you're patient and if you are having fun whilst doing this, it's amazing. So how do you manage all these things that you need to consider that I just talked about? So time management is an incredible thing that you will learn. If you don't have it right now, you will for sure learn. So balancing research, teaching, administration, and funding applications, all of this requires that you're very organized. This always improves. So you are, at the beginning, things might be, you think it's organized, but maybe it's not. So I think what happens is that it's very important that you set your goals and you set them in a time organized manner. So after a month, I would like to have this done. After 6 months, this is what I would like to have established already in the lab. And after 6 months, I go and I check, okay, did I manage? Did I not manage? And why did I not manage? Is this something that I can improve, or is this something external that I cannot do anything about it? And usually there's ways to mitigate when things take longer than what we expected. For example, I only got my mass spectrometer, which is a key technology in my lab last year. Like after a year, I started the lab. So what I did, I talked to very good collaborators of mine in the past, and I said, can you please measure the samples for us until we get our mass spec? So talk to people, reach out, and think of ways of mitigating these delays or this time waits. So there's always ways of getting around. Of course, it's not going to be as fast in the beginning, but I think there's always ways to get around. And another thing is delegating and not micromanaging. I think this is a bit hard when you're really starting your lab from scratch, because in the very beginning, you need to make sure that everyone is on the same page. Like Clinton mentioned, we also now have very shared protocols, shared folders, where people really get to know how to do things in a consistent way, in a reproducible way, because you can easily see that if everyone in the lab starts doing experiments their own way, then this is a mess. And this actually doesn't go in line with good lab practice and good science, because protocols need to exist and people need to do things in a consistent way. So in the beginning, as people are starting to join your lab, you need to make sure that they learn, or that the protocols are really passed on in an effective way. Now I feel that my lab is getting more and more independent, but in the beginning there was a lot of meetings and checking that everything was done in a very consistent way. So budget management, again, you need to know that there's different budgets. You will get third-party funding, and so they operate in a different way. There's institutional overhead costs. I guess I'm lucky that I'm in this research institute, and we have a lot of support regarding budget management. We have an entire team for that. They do advise us at multiple levels. They do explain all the differences between different budgets. So for me, this has been actually a relatively easy way to navigate, because I do get a lot of advice from our team, but I do know this is not the case for other institutions. So do make sure that you talk with as many people as possible to get to know how to navigate your budget. And then securing grant funding and networking. And I guess this answers the question how to maintain your lab and how to expand your lab. So I think networking is at
Clara,
the end.
if we could wrap up soon.
Yeah, so this is just one more. Yeah. So I think it's just like you really need to know what are the different fundings in your country. So in Germany, actually, it's quite well positioned for funding. And I do think that collaborations and networking really help also to know the different funding bodies and to apply together for funding. And building a positive lab culture, it's very important, and mentoring. So I think this also is something that you need to consider when you start your lab, how you want to set the culture in your lab. And institutional, this is my last slide. So institutional politics, you also need to know. And again, here you need to talk with as many people in your institute as possible, so you get a good overview. And this really helps navigate things in a much easier and faster way. And one last piece of advice is really being flexible. So you have your long-term vision, but we know that these things won't go all the time as planned. Timelines will be different, so be adaptable. And I think the most important thing is having fun whilst you start your own lab. There's a lot of things to consider, but it's also great fun. And with this, I would like to thank Big thanks to all my mentors, lab members, and everyone who actually helped me getting here, and you for your attention. If you do have questions that cannot be managed during the time, then please email me and I'm happy to be in touch. And that's it.
Thank you very much, Clara. That was really nice and see another take towards starting a group in a research institute. So we now welcome questions from everyone. So just use the box on the side if you want to ask one. And so now I'll just go through a few of them that we already have. So first one would be for Clinton. So we have Sanin asking about the time that you spent as a postdoc. So mentioning that you spent 7 years as a postdoc. And so when did you think that it was the right time to become independent? And what if your boss at the time would tell you that maybe you're not ready?
Okay, yeah, it's a good question. Thank you. I think when I became, I think privilege here, long postdoc, I had, you know, my boss at the time was happy to keep me on for a long time, so I had that security. So for me, it was really when I felt ready to manage people is when I thought, okay, I can start managing a lab and applying for these sorts of things. I guess if my boss had told me that I wasn't ready, it depends. It depends what their reasons are for thinking this, but I might have gone ahead anyway, or maybe not. I think that just depends. And I think I had a good, strong relationship with my postdoc boss, and that really helped, which I know is not everyone has.
Okay, thank you. Last one for Clara now. So Margareta Misha is asking about the choice of your mentor. So would you go for someone who's in your past current institution, the future one, or what is the general consideration, I guess?
So that's a very good question, because sometimes we think that we only need to have one mentor. I actually had several mentors, and how did I choose them? So I choose people at different career stages. I had people who had just started their own lab. I had really senior people. I also had like female mentors, male mentors, because the way they see things are different, but also what they had to go through was different. So it really is, I would say, choose in a diverse way, and that's it. So, and I think it will cover quite a lot of the questions that you will have.
I'll carry on with the questions from Andre Garlick to Clinton. So how did you choose your host departments, and did you consider different institutions? What would be best? How do you make that choice?
So for me, there were a certain number of lines we had. So I already mentioned that me and my wife wanted to stay in the UK. And then of the institutes, we really wanted to stay outside of London as well for, you know, it just wasn't our bag. And then of the remaining institutes, we looked for places with the techniques and the expertise that I required for the projects. That also my wife would be happy living in the locations too. Out of those, we had Oxford, Cambridge. We thought a while about a couple of Scottish institutes as well, and maybe York, but we eventually settled on Oxford. But it was really led by the application and by our life circumstance, sort of hand in hand.
Okay, thank you. And now we have a question from Paul Herrera, who's a postdoc in Malta. And so a question for each of you. So let's start with Clara. So after your 5 to 9 possible years at the Institute, what's your plan? Do you want to find professorship at university?
Yes, so that's the plan. I don't know if I will continue in Germany, because as I said, the funding perspectives are usually good in Germany, but permanent positions in Germany are not so good. The landscape there is a bit more tricky. So yeah, I'm just focused now on my science, getting, you know, the best science out of it, and then thinking, but yes, I would like to stay in academia and apply for positions. I don't know where at the moment. I really don't know.
Thank you. And so to Clinton, still from Paul, there was, do you have any pressure from Institute as to how many papers a year you need to publish and these kind of things?
So fortunately not directly, although in the UK we have the REF, of course, so I do feel some pressure there, and also some internal pressure as well. Not direct pressure, but that's probably because they know I will exert my own pressure on myself, perhaps, and maybe they would manage other group leaders differently.
Okay, and maybe we'll go for 2 last very quick questions. One from Julia, asking about how much time you would actually put aside to have preliminary data for your applications. So I'm not sure how much preliminary data you need for preparing for the research institute versus the fellowships. So enlighten us.
I mean, maybe I can just tip in for a research institute. If you're not applying for a fellowship, if you're applying for a position, you don't necessarily need any preliminary data. So it's very different for a fellowship. I think so. It's very different. I didn't go with a fellowship. I actually applied for research institutes, and I didn't have to show any preliminary data. So yeah.
Okay.
So maybe.
I think on my side, it really depends. That's a really annoying answer, but you know, I think if I was proposing a program of research closer to what I'd done in my postdoc, the barrier and the amount of preliminary research I will have had to show would have been less. In reality, I spent about 3 to 6 months collecting preliminary data, but this was very much on the side of my main postdoc project, which led to my last publication. It's a hard question to answer, I'd say, but hopefully that gives you a bit of insight.
And so last question from Charlotte Hutchins, who's asking whether either of you has supervised PhD students during your postdoc, and whether that would actually help you to supervise staff now as a group leader. So let's start with Clara.
Oh, sorry, supervising students as a postdoc helps supervising later on. Is that the question? I'm sorry, I was helps a bit, but not necessarily. And I guess this is because you will get a team with very different personalities. And I guess I stress that a lot because this is indeed what I find the most challenging. It's fun, but it is challenging to manage very different people and with different expectations, with different motivations or motivation level. And so it's very different. I supervised a lot of students in the past as a postdoc, as a PhD student, and it's very different. So okay. I can tell you.
How about you, Clinton?
I would say similar. It's different, but I think managing students at the postdoc level is good. I mean, it's a start, right? But as Clara was saying, I think managing is the key term. Ultimately now, you know, I am directly responsible for all members of my lab. They have different personalities as well, and that comes with an additional level. But supervising students at the postdoc level is for sure a great start.
Okay. Well, I think it's going to be time to wrap up. So thank you everyone for attending, and thank you very much to the speakers. We can continue the conversation online, so you can also follow at Biochemsoc on Blue Sky and X. So we're currently welcoming submissions for speakers to present their work at the 2025 ECR webinar series. So if you'd like to take the opportunity to share your latest findings, please consider submitting an abstract by 24th of February. So do it quick. So you can visit the website for more details. So all of our webinars that are listed on the website, any that you might have missed, are online. So please feel free to visit the website or YouTube channel. And the recording for today's webinar will be available within the next couple of weeks. And so finally, I'd just like to highlight that it's really important to stay connected, engage with our community of molecular bioscientists. In this respect, actually from a personal point of view, I think the Biochemical Society is doing an absolutely amazing job as connecting us in terms of organizing workshops, meetings, courses. They can also offer different types of grants and bursaries, access to different journals, and so there are plenty of benefits to being part of Biochemical Society. So please become a member if you're not already. And you can find much more details onto our website if you're interested. And with this, thank you very much to everyone, and goodbye. Thank you.
Bye.
Thank you.
Thank you.
Thank you.